Very good afternoon, friends. Myself, S M Moholkar. Today I am going to present homology modeling and ab initio modeling. Before that, we already heard about what is the word homology and where we use that. We are using that in the biological manners. Today I will going to tell you the learning outcomes in that first one in silico protein modeling as well as the available steps and its procedure, how we perform the homology modeling. And second one is the ab initio modeling with the different tools, what are the web tools or offline tools available for the homology modelings as well as an ab initio modelings. What is mean by homology? Homology is the range between similarity plus identity. Similarity means which is reaching to the 100%, and identity means the exact 100. That combines the homology. Why we doing the homology modelings? Uh, we are doing the homology modelings because they are made up of X-ray diffractions, in that only a small number of proteins can be made to from the crystals. A crystal is not the protein's native environment, very time consuming. Second method is the NMR distance measurement. This method is generally look at an isolated proteins rather than the proteins complexes, and it is the time consuming methods. Why does homology modeling? It is oftenly called as an comparative modeling also and knowledge based modeling also. In that proteins have the limited number of folds. The structure of a new protein can be resembles known as a fold even within no apparent sequence similarity. Structural information is always of great assistance in the study of protein functions, same dynamics, interactions with the ligands and the other protein contents. Uh, there are some principles and techniques using in the homology modeling. In that first one, development of a molecular biology. In that first one, rapid identification, isolations of a sequence, gene sequences. What are the problems during the homology modeling? Time consuming task to obtain the 3D structure of a protein. It takes the more times. Second strategy is in structural biology to develop the models of a protein when the constraints from the X-ray diffraction or NMR are not available. These two are the practical methods or experimental methods. Next, homology modeling is the method that can be applied to the generate reasonable models of the protein structures. Next, we are going to introduce the homology modeling. In that, homology modeling is used to predict the theory structure of unknown proteins based on the known structure of a similar proteins. During the evolution, sequence changes much faster than the structure. It is possible to identify 3D structure by looking a molecule some sequence identity. This method builds an atomic model based on the experimental structures. We already seen that NMR method and XRD methods. Then more than 40% with the target molecules. Modeling structures with less than 40% deployment similarity would result in the less reliable models, and hence it is ignored in the homology modeling. The known structure is called as a template and unknown structure is called as a target. Then homology modeling concept, how it involves. We are getting the unknown structures with the known called as a template, and known structure is known as a target. We are going to the sequence alignments that is available in the some sequence databases like Uniprot KB, Swissprot KB databases that going to the structure models. Database approaches of homology modeling. As per June 2000, 12,500 protein structures have been deposited into the PDB, that is RCSB PDB data bank, and 86,500 protein sequence entries were contained in the Swissprot protein sequence databases. This is the ratio of 1:7, and relatively few structures are the known. The number of sequences will be increased much faster than the number of structures due to the advance in these sequencing processes. How good can homology modeling be? sequence identity, we are going to the sequence identity, that is 60 to 100% is going to the method NMR substrate specificity. 30 to 60 is the molecular replacement in the crystallographic, that is XRD method. Supported site mutagenesis through the visualization, and less than 30% is the serious errors. It causes the errors during the homology modeling. Next, how we getting the homology modelings? By using structure predictions. In that first one, identifying the related structures called as an templates. Then we are going to template selects and align the sequence with a template structure. It is called as an target template alignments. Next, further, we are going to build a model for the target using the information of template structure. Then evaluate the model, and we are going to model and that check with exact match, and where this is, hence the process is going to end. Then further, this homology modeling is divided into three basic types. First one, basic modeling. In that model a sequence with a high identity to a template. Second type is the advanced modeling, model based on the multiple templates and bound to a ligand. Third one is the iterative modeling, increase the accuracy of the modeling exercise by iterating searches, that is repetitions. There are majorly we are using four types, that is template selection, target template alignment, model constructions, and model assessments. Rather than these four types, we are getting 7 more steps. First one, template recognition and initial alignment, we call as a target template alignment. Second, alignment corrections, backbone generations, loop modeling, sidechain modeling, model optimizations, model validation. And next, how we going to select the template in the homology modeling? In this step, we are comparing the sequence with the unknown structures with the known structures stored in the PDB data, protein data bank. A search with a blast against with the database will give the list known protein structures that match sequences. The accuracy of the structures generated by the homology modeling is highly dependent on the sequence identity between the target template. Above 50% sequence identity models tend to be reliable. In the 30 to 50% identity range, errors can be more severe and are often located at the loops. This is an online server Swiss model for doing the structure-based homology modeling. In that, we are first selecting the sequences, and when we clicking to the build a template, it makes that it getting the templates. These are some templates which are matching. Second step, target template alignment. It is possible that the alignment has to correct or change residues like alanine, glutamic acid is possible, but unlikely happen to the hydrophobic core, so our alanine and glutamic cannot be aligned. Using a multiple sequence alignment programs, we call that as a cluster W or cluster omega, the residue and the properties that have been conserved can be found. Second one, alignment, the most important step. Alignment of a low homology sequences can be improved using the second structure predictions, align, model, realign, and remodel it. We are getting template looking like this. We are getting this on the identity is first template getting the 100% identity in that, second also 100% identity, where ligand that molecule worked as a ligand also. So this is made up of X-ray crystallographic method, and also we are seeing their experimental methods, that is 2.3 Armstrong in it. This is a target template alignment, how it happens in the softwares. This is a tool, faster tool. We are getting the input target template alignment formats. In the target template alignment, we are using FASTA, PR, cluster W, PhyLib, MSF formats. These are required for the homology modelings. Third step is the model constructions. In that, assessment of homology models without the reference to target structure usually performed with the two methods in that. First one is the statistical potential and second one is the physical based energy calculations. In statistical potential are the empirical methods based on the observed residue contact frequencies among proteins known as structures in the PDB. Statistical potentials are more computationally efficient than the energy calculations. Physics based energy calculations aim to capture the interatomic interactions that are physically responsible for the protein stability in solutions, especially van der Waals and electrostatic interactions. We are saving that molecule in the form of PDB format, MMCF, DFUR format, model or PDB format to display that, and we are saving that model and look forward in the RASMOL tool also. See, this is an 3D structure of a molecule that I have getting during the homology modeling. Is there any tool for or databases rather than the Swiss model for homology modeling? Okay. There are few methods like Infold, RaptorX, Biscuit, Caps, CVHL, Spread 3D, and one more is their tool Fire that is also used for the homology modeling. There are some modeler and there are Python based basic modelings, and iTaser is also an ab initio modeling. There are some software for the homology modeling, Swiss model, modeler, ASPI. Then some errors occur in the modelings like a sidechain packing or distortion shifts, no templates. Errors in the homology modeling are like misalignments, incorrect templates, detection by using ProCheck, what if. These are two tools for we are getting the exact detection of homology modelings. Thank you.